Hello everyone, and welcome to phyloseminar.org. Today is the last talk of a series of 3 talks honoring the late Carl Woese, who revolutionized our understanding of the microbial world via molecular phylogenetics. Our first talk was from Norm Pace, then we had Ed DeLong, and today we have Phil Hugenholtz. We're very, very lucky to have Phil Hugenholtz speaking today, another one of the Norm Pace diaspora. He's published widely on microbial genomics and as well on methods to learn from genomes, and has a remarkable amount of expertise in the microbiology of wastewater treatment plants. He's well known for what has been known first as the Hugenholtz taxonomy and now is known as the green genes taxonomy, and it continues to evolve using the relationships inferred from genetic data. In this way, he is propagating the conceptual framework started by Woese. On a personal note, I should say that Phil, we've had sort of the most trouble I've had on any phyloseminar so far with technology, but he has suffered
No,
through
actually,
it.
what I'm doing is I'm dragging you forward by forcing you to do CVO, right?
Exactly.
So I'm doing you a favor, really. No need to
You're
thank
doing
me.
me a huge favor.
And it seems to be working now.
Yeah, so go ahead and take it away, Phil.
All right. Well, thanks for that very kind introduction, Eric. So I watched Norm Pace's and Ed DeLong's talks this week. I'd watched bits of them, but I thought I'd better watch the whole thing so I don't repeat what they're saying. And they together did a fabulous introduction to the beginnings of metagenomics and Carl's contribution. So I won't repeat that. I might just give a little bit of a personal perspective on a couple of pieces of it. So the outline, I'll give a quick recap of the 16S-based view of life and the importance of culture independence for improving that view, but I would definitely recommend looking at Norm and Ed's talks because they give a fabulous history to that. Then I'll talk about the challenges and limitations with the 16S tree of life, and then move on to how we can address those limitations by moving from gene to genome resolution of evolution. And then I will give a case study, sort of giving some examples of how we can get that genome resolution with candidate phylum TM7, one of my favorite novel phylogenetic bacterial groups. And if time permits, which I suspect it won't, I will briefly talk about a soon-to-be published JGI single-cell genome study, which is also addressing this genome tree resolution of life. Okay, so Carl Woese was prescient. He had the extraordinary foresight to identify the subunits of the ribosome as molecular chronometers upon which an objective tree of life could be based. And so he was there at the very beginning, as Norm and Ed both pointed out, at the very beginnings of sequencing technology, and he took it. He took that in hand very early on, and made amazing contributions to the field, upon which my career rests and many other people's careers rest. So I'll just also highlight this amazing paper that he wrote, a review in 1987. Both Norm and Ed pointed it out, but I'll give my personal perspective on this. So I was in the 3rd year of my science bachelor's undergraduate at the time that this paper came out, and it was assigned reading. And my first thought was, I cannot believe they have given us a 50-page review as assigned reading. I said, this is just going to be a nightmare. And so I started reading it, and it was like, you know, one of those moments when the clouds part. I was vaguely aware of what Woese had done at that point. It was still fairly new, and it wasn't making its way into the teaching curriculum in any great way. I was in Australia at that point. But it was an epiphany, I have to say. First of all, he writes beautifully, and so it's very easy to read 50 pages, and it was all just laid out for you to see. And this was on the back of, I'd done a 3rd year numerical taxonomy course on how to classify bacteria, and I just wasn't getting that stuff. I mean, I catalase, oxidase, I couldn't keep any of it in my head properly. It was just a big gamish. And when I saw this, and I saw this nice, clean, objective way of classifying bacteria, it's the framework, I just went, that's it. That's the way you do it. And I recognized early on that was going to be a much greater friend to me than the classical phenotypic testing. Not that I should run that down too much, by the way, because the classical phenotypic testing, there's a huge history and a lot of work went into that. So this is one of the figures showing the bacterial domain from that 50-page review. And the one thing in common about all of these organisms that went in to make this 16S-based tree is that they all were cultured. And as Norm and Ed both pointed out, the great majority of organisms that you can observe under the microscope in the wild don't end up on a plate. The counts don't match, and so that was formalized in Staley and Konopka's 1985 paper where they termed it the Great Plate Count Anomaly. And so, as has already been pointed out in great detail, very entertaining detail by Norm and Ed, Norm built upon that framework that Carl developed and applied a culture-independent approach using those ribosomal RNAs as markers, so I don't need to dwell on that. So I was fortunate enough to do a postdoc in Norm Pace's group in the early 90s, and I had the nice job of doing one of these search numbers, culture-independent surveys of one of the hot springs, Obsidian Pool, and I sequenced a couple of hundred clones, and out of that a whole bunch of novel lineages emerged called OP. And this was a paper that came from a few years later in 1998. And each of these wedges represents a major line of descent within the bacterial domain. And in terms of sequence divergence of ribosomal RNA, each of these has as much divergence as the corresponding genes in plants, animals, and fungi combined. And so all of the blue lineages have one or more cultivated representatives, and the red lineages at that time had no cultivated lineages. And I remember really being blown away by this. I think to myself, how can one postdoc go in and have this small project, go and grab a handful of sediment and sequence it and come out and find all this uncultured diversity, and every postdoc PhD student doing this was finding this level of diversity. And I like to tell the students, and the way I think of this is if you were a geographer and you had been studying, the fjords in Norway your whole career, and then suddenly somebody says, hey, guess what we just found? We found the Americas, and there's Australia, and there's all these other continents we've never even seen before. That's what this is like. It's like this incredible amount of diversity, which going in and just applying the ribosomal RNA genes, we had groped the outlines of. So I was incredibly taken with that. And like many others, I said, that's my career sitting right there. That'll be what I'll be doing for the next 20 years, looking at that, and that's indeed what I have been doing for that length of time. So this was from 1998. That's a good 15 years ago now. So in 2012, we took another census, and using a fairly conservative estimate where we said, there has to be at least 20 environmental sequences representing this candidate group. We estimated there's about 60 bacterial phyla, and half of those are uncultured or candidate phyla. Norm showed a picture yesterday, the increase in the numbers, and he was estimating about 100. If you reduce the stringency on how you define a candidate phylum, the numbers go up to 100 or even 120. So this is showing how the sequences have been accumulating over time from the year 2000 to 2012. And the dark green line is showing 16S sequences that are coming from isolates, and the light green line showing sequences that are coming from environmental sequences, so from environments, from these surveys that Norm pioneered. So this is not a count of numbers of sequences on the y-axis. This is an indication of the cumulative phylogenetic diversity. So as you make a tree and you put another sequence in, you're counting how much novel sequence diversity is it adding to the tree. So what you can see is that starting off in 2000, about 86% of the described diversity at that point was coming from isolate sequences, and only 15% or so was coming from these environmental sequences. It's in early days. Now, that's completely reversed. Now, these days, you've got the opposite. You've got about 86% of the diversity coming from the environmental sequences, only 14 slowly accruing from the isolates. Now, I might also point out that these are near-complete 16S sequences, 1,200 base pairs or more that are in the databases. There's about 1 million sequences. In 2012, this is representing about 1 million sequences. And so you can see how, just looking at the phylogenetic diversity, there's much, much greater phylogenetic diversity has been discovered within the last decade, most of it in this uncultured fraction, this microbial dark matter. So Norm talked about challenges to resolving the 16S tree of life, and part of that is providing a sound reference taxonomy upon which a lot of the ecological work is based. And so there are several databases out there that are dedicated to looking at 16S and making that the basis, the 16S phylogeny as the basis of the taxonomy for the bacteria and archaea. I've only put 2 down here. Green genes, because I'm directly involved in that. I curate that database in my spare time. You know, it's an obsessive compulsive disorder, but there you go. And then the silver database, which is a database, there's another person over there, the whole team that does it, but another person that's my equivalent that also has obsessive compulsive disorder and likes to curate databases. And so Norm was talking about short reads, the pyrobabble being an issue with the taxonomy. That's certainly the case. You would not take these short 200 or 300 base pair reads and do de novo trees, because the phylogenies that come out of those short reads, there's not enough signal, are very poor. And the amount of pyrobabble, we checked this morning, I'm not sure I really like the term pyrobabble, but just pyro tags or pyro sequences in the SRA at NCBI is about 40 million. No, I'm sorry, 400 million. 400 million sequences currently. So 400 times the amount of full-length 16S sequences upon which the taxonomy is based. And I think, you know, you can, as long as your underlying taxonomy is sound, you can classify these pyrotags against it and make good ecological inferences. So that's where I see the use of those reads, and it's a screening tool, really. Now, maybe more importantly is just the sheer numbers of sequences. So even not including those partial or short pyrotag sequences, and even shorter ones if you look at Illumina-based 16S tags, the full length is 1 million sequences. That's a challenge if you want to do a phylogeny. So Silver, what they do, they had a starting tree, a 16S, and then they use a parsimony insertion tool to put new sequences into the tree, and then they base their taxonomy of that. At Green Genes, we've tried to do de novo trees, and so there have been tools developing out there for being able to make very large taxonomies from hundreds of thousands of sequences, and that's what we've been using. And then a big enemy, in fact, I'd say enemy number one, if you're trying to make de novo phylogenies upon which to base taxonomy, are chimeras. And these are an artifact generated during these environmental surveys, and I'll talk about that in just a second. But ultimately, the ultimate enemy, or the ultimate problem with the resolution of the 16S tree of life is the actual marker gene itself. It's a fabulous molecular chronometer, as we know, but it's only 1,500 nucleotides long. So you're reaching the resolution limits of what a single marker gene can do. I mean, you're asking a lot, actually. You're asking, tell me about 3 billion years of evolution across 1,500 nucleotides. So that's an ultimate obstacle to getting a highly resolved tree of life. Now, just revisiting the chimeras, because I really hate chimeras with a passion. I loathe them, so I'll spend a second talking about them. So what happens when you have a number of templates within environmental DNA and you go to PCR amplify it up using broadly conserved primers, is that some subset of the sequences have an aborted extension. So here's your template sequence here, and you start to do your amplification, and it falls off, polymerase falls off, and so you're left with this partial 16S sequence from one organism. And because of the high conservation of the 16S RNA, which is one of its strengths, it can misprime. It only has to have a sequence identity at the 3' end, maybe as little as 10 bases, onto another template and can continue extending. So now you have a chimera of two different members of that community, and you're going to stick that into a tree, and it's going to absolutely screw up your phylogeny, or it can, depending on how different the two parents are. So this has been known for a long time, and this is it's a well-recognized issue, and it's been recognized since the 90s, pretty much since the beginning of doing environmental surveys. And I put out a little piece in the early 2000s, sort of where I was manually checking for this as part of my curation tasks at Green Genes. And this was back in the days, remember from the chart, where we had much fewer sequences and only 15% of them or so were actually environmental sequences. And it was kind of alarming, the number of chimeric 16S sequences that were slipping through the cracks. So I'm here to tell you today, with the tables turned, and you've got a huge, what did we say, 850,000 full-length 16S sequences. There's a ton of chimeras still in the databases, and this poses a major challenge to coming up with a sound tree and a sound taxonomy. So there's no way to completely prevent chimera formation. There have been ways that you can reduce it, and there's no way to perfectly detect chimeras. So I've been involved in trying to make tools for detecting chimeras. Other people have been involved, and if you apply those tools to the same data set, you get somewhat overlap, but you don't get a perfect overlap. You cannot perfectly identify the chimeras, and sometimes you detect them manually, and you go, how on earth did the algorithm not pick that up? So partly that's because if you have multiple chimeras of multiple different levels of recombination between different levels, that makes it very difficult. You need a good reference set that are true and not chimeric. And one of the things we used to use for chimera detection was the idea, well, if I have a sequence that's about 97% or more identical to another sequence produced by another lab on the other side of the world, they did their PCR over there, so there's no way that there was crosstalk between the two studies. That must be a sound identifier of a non-chimeric sequence. No. So you can actually get reproducible chimeras occurring between similar habitats. So for example, if you're looking at the gut, we know that the gut's dominated by Bacteroides and Clostridia. Well, you can actually get reproducible chimeras between those two groups, which are in different phyla. And so you see in the databases sequences that are close to each other. They have a chimeric junction that are within 15 or 20 bases of each other, and they form what looks like a cluster, but they're chimeras, and those chimeras can drag large groups into the wrong parts of the tree, so you can actually drag in a chunk of the Bacteroides into the firmicutes. That's why I hate these things so much. So what's the solution then? So the obvious solution is to supersede the 16S trees with genome trees. So you don't get the PCR-induced chimeras. You might get natural chimeras from evolution. You get higher phylogenetic resolution because now you've got the whole genome to play with rather than one gene. And there's a whole huge story there in terms of looking at what part of the genome you'd look at because of the lateral transfer, which is a natural part of evolution. And then, that's just the tip of the iceberg in terms of making trees, but you're essentially getting the genome sequences, you're coloring in the outlines that 16S provides. So 16S provides you with the framework, and now you're coloring it in. So, obvious things like what's the functional potential, you can determine that from the whole genome. You can overlay routes of lateral gene transfer on that framework, and et cetera, et cetera, et cetera. There's many, many things you can do with genomes. Now, as Ed pointed out, Carl Woese was a very early adopter of microbial genomics. If you read, I couldn't fit the abstract in here, but he said, I don't really care about the human genome. That's going to be a sideshow in the long run. Where it's really going to be happening is in the microbial genomics. And he was really, in that very early days of genome sequencing, he was pushing for getting a representational coverage of the tree of life through genome sequencing. And he is dead right, I think. So the downside currently is that the genome database is much smaller than the 16S database. We're looking at maybe, I haven't checked the Genomes Online database that Nikos Kopidis curates, but it's, thousands, 3, 4, 5,000, as opposed to full-length 16S 1 million, as opposed to total 16S 500 million. And worse than that, those sequences are not representative of microbial diversity, very skewed, in fact. So there's been some heroic efforts to address this system. There was a genomic encyclopedia of bacteria and archaea, which was a joint effort between the Joint Genome Institute in California and the DSMZ, the culture collection in Germany. And that's Hans-Peter Klenk and Nikos Kopidis were major drivers of this, and Jonathan Eisen and Eddie Rubin. And the idea was just systematically use the 16S tree of life to identify organisms, isolates within the culture collection that need to be sequenced in order to get a more representational coverage of the tree of life. And so the pilot study was published in 2009 with 56 genomes. That's an ongoing study. I think it's in its 3rd iteration now, and there are a couple hundred genomes, which have been, I think, a fantastic research to the scientific community and are starting to balance out the coverage. But keep in mind, that picture I showed before, you're really tapping into 14% of the total phylogenetic diversity in microbial diversity only if you're looking at these isolates. So what you really need to do is to get into this light grey stuff, all this environmental sequence for which we don't have isolates. So how do you get at this stuff, and how do you do it fast? You want to be filling in that tree as quick as possible with genomes so you can get that representational picture. So one way to do it, which has been discussed by both Norm and Ed, is to use metagenomics. So that's the natural progression of 16S-based culture-independent analysis of communities. You just go in and you shotgun sequence the community, and it's like genomics, except that you assemble the reads into contiguous pieces, and then you call the genes on, you annotate the genes on those pieces, but it has the additional step of this binning. So if you want to get back genome sequences of species, or in this case, because it's coming from environmental populations, you have to be able to identify those anonymous contigs and put them back into a genome. And that's been pretty tricky and slow going, and so there's been some, very good examples where you can get back, say, a dominant population, you get plenty of sequence data back for that, and you can assemble a near complete, or in some cases a complete genome straight out of the environmental gamish. But traditionally, it's been thought that, if you were to be aiming for some of the more exotic species, you never see them as dominant members of communities. So how do you get at that using metagenomics? So I'm going to cut to the case study now with TM7 as an example of the progression of the methods and how these genomes can be accessed. And this started with a study by Erko Stakkebrand. So Erko Stakkebrand was one of Carl Woese's students, and he's one of the guys that walked out of the lab after a hard day looking at those patterns with 10 millicuries of radioactivity under his fingernails. And so he's right in there at the very beginnings of the 16S-based environmental work, and in this relatively early stage from 1996, He was looking at a peat bog in Germany, and the German translation to TM as the moniker. And it started with this single sequence, which was termed TM7, and it wasn't sitting within any of the recognized lineages that were currently recognized by isolate, so it's just sitting out there by itself. And by 1998, we'd recognized that there was a few more sequences coming out of environmental surveys that it was labeled as an independent candidate phylum, and here it is here, TM7. And so, there's so much diversity to pick from. I was keen to start trying to get in and start understanding what that diversity was. So together with Linda Blackall and Jean Tyson, a student at that time, we went in and using the full thickness approach that Ed described, designing probes and primers that were specific for TM7. So by this stage, around 2000, there were maybe 50 sequences in the databases that belonged to this TM7, and from this we designed probes, and we went into one of our favorite pet environments, which is activated sludge, and applied these probes and uplit this fabulous, I remember the first time we saw it, couldn't believe our luck, this giant filament just stood out, boom, from the probes, and so there was a TM7 there right there. And so because this was a very conspicuous filament, it had a sheath, and it was the main large filament present, we could actually go in and look at, under electron microscopy, and this was work done by Rick Webb, and we could identify this sheath filament. So he looked at 200 and 197 were this sheath filament, and from that we could deduce that this TM7 morphotype had typical gram-positive or single-layer monoderm cell envelopes. So we're feeling, oh, that's pretty cool. We can deduce something about this organism we can't grow. And then this is a nice story. Ed DeLong was actually visiting us in Australia at that point, was visiting Australia, and I was talking about this in the talk, and I put up the full secondary structure of the TM7 16S sequence, and Ed was scratching his chin looking at it, says, there's something funny about that 16S sequence at around position 900. So you can tell how many 16S sequences he'd been looking at the time, and what he'd spotted, which is incredible because I didn't have that slide up for that long, he'd spotted that there was an atypical uracil in position 916 or something like that. I can't remember the exact number, 912 maybe. So this is a highly conserved part from which some of these primers are based, and he spotted this, and he said, normally see a uracil in archaea. And he said, furthermore, that's the basis of streptomycin resistance. So we had a hypothesis then that the TM7, unlike most bacteria, were streptomycin resistant. So we published this in 2001. I hope we credited Ed for that fantastic spotting that in that talk. I do it now publicly anyway. Good job, Ed. And so we had that, and then Per Nielsen in Denmark was using those probes that he'd come up with, and he was doing a micro-order radiography to see what type of carbon sources the TM7 filaments were capable of taking up. And what he saw was that they very, very great facility for taking up simple sugars, such as glucose and galactose. So just pointing out that even with no cultures, you can get a fair way in using the full cycle rRNA approach. So just quickly point out that TM7, I remember the paper actually, the one we wrote actually originally was rejected from Applied Environmental Microbiology because essentially the reviewer said, who cares? What's TM7? I don't understand what the significance of this is. And really there wasn't a huge amount of significance at that point. It was more curiosity driven. But as fate would have it, TM7 started to be recognized for human clinical relevance, and as soon as it involves humans, of course we want to study it. We don't care if it's in a hot spring or activated sludge, but the minute it's in humans, we're interested. So there's been a great number of publications now showing that some TM7 species are associated with inflammatory mucosal diseases, so they can get through the epithelium. And so there's some nice studies here. David Relman's group, for instance, Bruce Pastor, and they show that it's in the mouth, so you can get it associated. It's associated with subgingival plaque, and you get it in guts, so associated with things like inflammatory bowel disease. And so there've been some very nice studies where the microbial ecologists have been working in concert with immunologists and showing that the TM7 have significant associations with these inflammatory mucosal diseases. So by 2009, I wrote a little review together with Mircea Podar on TM7, and by that stage there were a few hundred sequences that were now belonging to TM7, and this was a sort of a summary. And so we had two major subdivisions defined based on the original analysis, and coming from a range of habitats. There were the clinical ones. The clinical ones were mainly found within subdivision 3, and so we still need to get to the point of how do we access the genome. So now we had a lot of information. You know, we had some said it's got a gram-positive cell envelope. It might be streptomycin resistant. It's important in clinical environments. Now we want to get to the genomes and find out, learn a lot more about them. So one way to go is to divide and conquer. So rather than just do metagenomics on the microbial community, go for an enriched population or even down to a single cell. And we know that TM7 is widespread in nature, but it's low abundance. So just doing bulk metagenomics was thought that's not going to be very successful and be able to pull out the TM7 genomes. So in fact, this was a kind of the poster child for some of the early single cell genome studies. And if you're reducing the complexity down to a single cell, your assembly problems go down because now you're assembling one organism rather than multiple, and your binning problems go down for the same reason. On the downside, you're getting much lower amount of starting DNA, so you need to have a whole genome amplification using things like multiple displacement strand amplification, and that has an associated set of problems. You get uneven amplification, so you get representational bias within the genome, and you also run a higher risk of contamination because you're starting with much lower biomass. So just point out here that in terms of TM7 specifically, Belinda Ferrari in 2005, having read about putative streptomycin, used streptomycin as a selective agent to try and enrich for TM7, and also used filtration. A filtration by a Japanese group showed that you could enrich for TM7. So the first, or when I talked about those relatively early studies on single-cell genomics, Steve Craik's group used their microfluidic chambers to get oral cells and identify them by 16S, and he found these filaments in there that would belong to TM7. And so he contacted me. I was working at the JGI at that time and said, "You've done something on TM7. Would you like to be involved in the genome analysis?" And I said, "Fantastic." So I got very excited when we got the first genome data back because it showed evidence that the TM7 had genes for both gram-positive and gram-negative cell envelopes. So I was just seeing the Nature and Science papers come up, you know, paradigm shifts, rah, rah, rah. But it turned out that the genome that came back had been contaminated with leptotrichia. Leptotrichia is a gram-negative organism belonging to the fusobacteria. It's common in the mouth. And so what we surmised is that this leptotrichia DNA had been piggybacking on the TM7 cell. And so there was our hybrid gram-negative, gram-positive signal. Still a very nice paper, by the way. Okay, and then another study around the same time, Martin Keller and Mircea Podar were looking at TM7s in soil, and they also had a little problem with contamination from Pseudomonas. They managed to, it was easy to sort out based on GC content, and they estimated that they got back 20% of a 3 million base pair TM7 genome. Some of the metabolic inferences were in concert between the clinical one that came out from the Marcy study and from this study, so there were some things in common, but in both cases the completeness of the genome was not great from the single cell study. So this is where I'll bring in, going back to metagenomics. So we revisited TM7 with metagenomics, but with deep metagenomics. So the basic premise here is that, you know, if you just sequence more deeply, and the sequencing technology has been ramping up, as everybody knows, over the last decade greatly, so you orders of magnitude more sequence. So if you just get more sequence, you're going to sequence deep enough that you can get enough sequence data to reconstitute genomes of low abundance organisms like TM7. And so Per Nielsen, who I mentioned before, did the Fishmar, and his student Mads Albertsen came and visited us here in Australia, and they had generated sequence data from activated sludge, and they were actually not after TM7s. They'd been doing a lot of probes and checking things out, so they knew TM7s were in this reactor, and they had used two DNA extraction protocols to try and get back DNA from a gram-positive organism they were interested in. They were thinking that the first method they'd used wasn't rigorous enough to get back the DNA from the organism. And that turned out to be very fortuitous, because essentially what happened is that the extraction, the two different extraction methods, pulled out DNA differentially from different populations. So let's see if I've got. So I'll show you how we can show this. So you can think of it like this. This is a plot showing the coverage of, so each of these dots represents a contig. The size of the contig is by the size of the circle, and the coverage is the read depth for each of those contigs. And so you can match up those contigs between the assemblies. They'll have slightly different coverage. So you can put them as a unique dot on this 2D plot. So I like to describe it in some ways to a molecular biologist. It's easier to understand. If you do a protein gel and you run it in one dimension, you get some separation of the proteins based on size. If you then apply the electricity at 90 degrees, you separate on a second axis, so you get higher resolution. So this was a nice approach to getting better separation of populations based on this differential coverage. So just to make that a little clearer, if you take multiple metagenomes of related samples with the same populations and you use different extraction procedures, as in this case, or you take it at different time points, or you take it at slightly different spatial points, anything that will result in slightly differing relative abundance of a given population, you can use that as a signature for a given population. So in this case, you see you've got the green population, 3 in the first sample, 1 in the second, and 2. And so it has this signature of coverage that you can use to help you bin the data. And this is called differential coverage binning. And so using differential coverage binning as the primary binning tool, Mass went into this sludge data set, and he could pull back a number of populations. And so each of these dots represents a contig again, and the size of the contig is the size of the dot. And so after you get your contigs back and you bin them into their basic bin, so the blue ones belong to one bin, green ones belong to another bin, you can start to do standard assembly tricks like use paired-in data to link those up. And so this is showing a number of populations linked up by paired-in data. And so in some cases, you've got very clear and highly assembled populations that fall out like this. In other cases, you've got these masses like this, where very small contigs, very complex graphs connecting them. What this is telling you is that this is from a population that has a lot of strain heterogeneity, so a lot of closely related ones, and that mucks up the assembly. And if your assembly's mucked up, then your the population genome you get back is very compromised. I'll come back to that very briefly in a minute. So after doing the primary differential coverage binning, you can refine that using other standard binning methods like sequence composition, and best hit to reference genomes, for instance, which I haven't got time to talk about. So you can refine the assembly, and Mass did this. And so he was able to get it to a point that many populations he could get back to a very good quality draft genome. So here's an example of a TM7 genome coming from that assembly, which could be broken down into 15 pieces. And the only uncertainty are repetitive elements within the genome that here with the higher coverage in red. And so this is a very, if you can get a genome back in 15 scaffolds, that's a very good quality draft genome. Now the amazing thing was that when you looked at the relative abundance of these TM7 genomes in the original sample, they're really low. So they actually range from a couple of percent down to 0.06%. So this is at the levels that you typically would not have thought that you could get at genomes using metagenomics. So that's very exciting. And these are very good. One of the first one that was at 2% was in a single scaffold, so it got back as a single piece. And one of the features that came out very early on about these genomes is that they're quite small, towards the lower end of the spectrum for free living organisms, 1 million base pairs. And at first we thought, oh, this must mean that we haven't got a complete assembly, but if you're using, looking at conserved marker genes, this indeed indicates that these are and assembly itself, if you can get it into one piece, tells you that you've got the bulk of the genome back. Are you still there, Eric? I'll assume you are and just keep going. I won't go for too much longer. So we could do a metabolic reconstruction of these sludge TM7s, and sure enough, the genes told us that they are indeed very facile at eating sugars, and so they have pathways for generating energy and carbon out of those sugars. And so we renamed the TM7 as saccharibacteria because of their predilection for sugars. And they also had genes that were indicative of gram-positive cell envelopes, which was the original inference from that original study. And so if you take the genes that maximally differentiate gram-positive from gram-negative organisms, so gram-negative organisms have lipopolysaccharide, and then put it up against TM7, here it is, you see that it's got the gene complement that speaks to a monoderm cell envelope. And so together with well-known gram-positives like Firmicutes actinobacteria. And this is a tree, genome tree, made from concatenated marker gene set, which places the TM7 within these gram-positive or monoderm lineages or atypical monoderm lineages. So that's all nice, close the loop. However, the sum total of all this work at the moment is that we've got partial genome sequences in this part of the tree from the soil study and the mouse study, and this is from the recent deep metagenomic sludge study. And so we still are a long way from even having representational coverage of the TM7 phylum. So there's a lot more work to do. Now what I'll quickly do a plug for GroopM, which is a tool developed to do differential coverage binning by Mike Immelfort and Jean Tyson's group here at the Australian Centre for Ecogenomics. And what you see here, each of the dots represents a contig, and they've been placed in an n-dimensional space according to their differential coverage signature. And the tool automatically goes in and identifies putative population genomes and then deletes them and spits out. So typically, from about 50 gigabases of data, you'll get dozens of population genomes coming back. So I point that out because it's to do with being able to tap into that microbial dark matter fast and get genomes. Hello? Have I just been talking to myself? I'm happy to do that. Anyway, it's recording. So what I might do, because it's already 9:45, I was going to talk about the, hey Eric. So keep going. I can't hear you now. 1 minute. Yep. Okay.
You disappeared for a bit, but I bet that your recording was fine.
I was recording it. Yes. So
okay.
I just soldiered on. So I'm going to wrap it up now. I just went through the rest of the TM7. It's recorded, and I think for interest of time and maybe having a few questions and basically on the other talks I've seen, I'll just cut to the chase now. I wasn't sure I'd get time to talk about this single cell study from Tanya Worker's group at JGI. Essentially, the bottom line is that even though I was a bit negative about the single cell genome studies of the TM7, Tanya has been working very hard to get over some of those problems and has applied it in a systematic fashion to tap into the uncultured microbial world. And she has pulled back 200, her group has pulled back 200 single-cell genomes, and they span 25 phyla. So that's very exciting. It's covering a lot of those groups that I've been interested in. And I was going to talk about tree, but I'm going to cut to the chase. If anybody's got a question, I'm happy to take about that. But there's been some developments there, a whole bunch of new names for a bunch of environmental lineages. And so we have some starting points for what their physiology and evolution is about. So I'll just finish by saying I think these are very exciting times that we live in, but we still have a monstrously long way to go in terms of articulating that genome tree of life. So if you first of all look at this inset box here, this is again on the y-axis the phylogenetic diversity, covered, and 16S by the way, covered by genome sequences. So if we look at which organisms have had their genome sequenced just as part of the genome sequencing history since the mid-90s, you have, and ranked by the coverage, you get about a PD of 200 here. And then this is the GBA, looking at the isolates where there was a systematic selection to try and improve that coverage of phylogenetic diversity, and you get the little bump. And then this is the study with the single-cell study from Tanya Woike's group, and you get another little bump. But then if you look at that in the context of how much is left to do, there's enough work for everybody to go around, I would say. We need another 20,000 genomes, and that'll get us maybe halfway up that diversity hump. Now, not just any 20,000 genomes, 20,000 genomes selected to maximize phylogenetic coverage. So, you know, I'm very, I wouldn't say now, oh, that's going to take 100 years to do that, because 10 years ago I would have said it would take 100 years to get where we are right now. So if we continue to ramp up at this speed and the methods keep developing, I think we really will have a truly objective view of the of evolution and the tree of life through genome sequencing. So now I'll just finish with this cartoon I found online of Carl Woese. It's a little underselling his contribution, but I can certainly identify he's got that urge. I've got that urge again. It's uncontrollable. I must classify. So classification taxonomy is viewed as a sort of a dry art, but in fact it's a cornerstone for everything on top of that. So I think that's a very good urge to give into. And also I think they were very unfair in the amount of hair they gave him, because he has a lovely full crop of hair, and so this was underselling him on many fronts, I would say. Okay, thank you. I'm happy to take any questions.